Introduction to DNA Microarray. DNA microarray, also known as DNA chip or gene chip, is a technology used to analyze the expression or presence of thousands of genes or DNA sequence in a sample. It is a powerful tool in molecular biology and genomics research, allowing scientists to study gene expression patterns, genetic variation, and other molecular interaction on a large scale. The basic principle of DNA microarray involves immobilizing single-stranded DNA fragments known as probes. These probes are designed to be complementary to specific DNA sequence of interest, such as genes or genetic markers. DNA microarrays can be used for various applications, including gene expression profiling, comparative genomic hybridization, single nucleotide polymorphism analysis, and genotyping. They have been instrumental in advancing research in fields like cancer biology, pharmacogenomics, developmental biology, and personalized medicine.
Let's continue with the DNA microarray techniques. DNA microarray techniques can be divided into 4 steps, which are isolation, hybridization, washing, and analysis. In isolation steps, it is required to have control and target mRNA to be isolated. The RNA is then converted into cDNA and labeled with fluorescent dye. There are 2 types of dye that are usually used in this isolation process, which are Cy3, a red fluorescent dye, and Cy5, a green fluorescent dye. In a hybridization process, the sample DNA is hybridized with complementary probe sequences on the array chip. DNA will strongly or weakly hybridize or not at all. To the washing process, the slice is then washed with buffer to remove any DNA that did not strongly hybridize to a probe. This will help to prevent cross-hybridization. Finally, analysis will be run as a final step of DNA microarray. The array chip is exposed to laser excitation to show the relative abundance of hybridized DNA in each target location. The fluorescent emission is measured using confocal microscope. There are 3 colors that may be emitted by the chip, which are red for up regulation, green for down regulation, and black.
DNA microarrays are a powerful tool for gene expression analysis, but they have several limitations. These include indirect measure of relative concentration. The signal measured at a given position on a microarray is not linearly proportional to concentration of the species hybridizing to the array. This means that the signal is only accurate over a limited range of concentrations. The second one is difficult to design arrays for complex genomes. It is difficult to design arrays that will uniquely detect every exon or gene in genomes with multiple related genes. This is especially true for genomes with a high degree of The last one is can only detect sequence that the array was designed to detect. If the solution being hybridized to the array contain RNA or DNA species for which there is no complementary sequence on the array, those species will not be detected. This means that genes that have not yet been annotated in the genome or non-coding RNAs that aren't yet discovered as expressed will not be represented on the array.
The solution used in the DNA microarray is called the hybridization solution. It is a mixture of chemicals that helps to promote the hybridization on the labeled DNA probes to the DNA on the microarray. The hybridization solution typically contains the following components such as sodium dodecyl sulfate, sodium chloride, sodium citrate, dextran sulfate, and fluorescent dyes. The hybridization solution is applied to the microarray after the DNA probes have been labeled. The microarray is then incubated at a specific temperature for a period of time, allowing the labeled DNA probes to hybridize to the DNA on the microarray. After hybridization, the microarray is washed to remove any unbound DNA probes. The signal from the hybridized DNA probe is then detected using a scanner. The hybridization solution is an essential component of the DNA microarray technology. It helps to ensure that the DNA probes hybridize to the DNA on the microarray in a specific and reproducible manner. This allows for the accurate measurement of gene expression levels, which is the primary application of the DNA microarrays. Here are some recommendations or additional tips for using the DNA microarrays. First is by choosing the right microarray platform. There are many different microarray platforms available, each with its own strengths and weaknesses. It is important to choose a platform that is appropriate for your specific research goals. Next is by using high-quality RNA samples. The quality of your RNA samples will have a significant impact on the accuracy of your microarray results. Make sure to use RNA samples that are free of contaminants and that have been properly stored. The next recommendation is by using appropriate statistical methods. There are a variety of statistical methods available for analyzing microarray data. It is important to choose a method that is appropriate for your specific research goals. In addition, always be aware of the limitations of microarrays. Microarrays are a powerful tool, but they have some limitations. For example, microarrays can be sensitive to noise and can be difficult to interpret. It is important to be aware of these limitations when designing and interpreting microarray experiments.
Conclusion: DNA microarray is a tool to determine DNA from a particular individual contains a mutation in genes. The principle is complementary sequence will bind to each other. It is done by base pairing rules. The unknown DNA molecules are cut into fragments by RE. Then, fluorescent markers are attached to these DNA fragments. These are then allowed to react with probes of the DNA chips. Hence, DNA microarray helps us a lot in clinical and also research. It helps us learn about differences in gene expression between a healthy cell and a cancer cell.